Hi, and welcome back to the second video for Chapter 17. Here we're going to look at cloning and genetic engineering. Our next technique is molecular cloning, where we're trying to clone some kind of gene of interest. And as we've seen in the lab, to do so, we need some kind of plasmid or vector that is going to be used to hold our gene of interest so that we can deliver it into a bacterial cell and it can make many copies of the gene. So we've seen before that plasmids are small circular DNA molecules that replicate independently of the main chromosome found in bacteria. They work really well because they have a region called a multiple cloning site that contains restriction sites. Restriction sites are specific nucleotide sequences that are cut by restriction endonucleases, which are also known as restriction enzymes. When restriction enzymes cut at these specific sites, and each restriction enzyme has a specific sequence that it will cut at, it can linearize the plasmid or open up the plasmid. And at the ends of the cut sites, we have sticky ends, which are single-stranded ends that will bind to other complementary single-stranded ends. If I have my gene of interest, let's say this is my human DNA and I have some kind of gene of interest, and I happen to have the same restriction site flanking either end of the gene of interest, I can use one restriction enzyme to cut here and cut this out, and the same restriction enzyme to cut open the plasmid. That means the sticky ends flanking the gene and at the ends of the plasmid over here will be complementary, which will allow the two pieces or two molecules of DNA, the plasmid and the gene of interest, to bind together. To really stick them together and hold them in place, however, we need DNA ligase, an enzyme that almost acts like glue to permanently join the gene to the fragment of plasmid. Here's an example from our textbook that displays a really common method of cloning of genes. So here I have my plasmid on the right, and I can see my multiple cloning site, which contains a restriction site that will be recognized by a specific restriction enzyme. And it also happens to be that the restriction site is located within a gene called the LAC-Z gene. If you cut open the plasmid at that particular restriction site, you will disrupt the LAC-Z gene. And LAC-Z is a gene that's necessary for the bacteria to metabolize lactose. I can see the plasmid also has an ampicillin resistance gene over here on the other side. So that's my plasmid or vector. And on the left, I can see my foreign DNA. Let's say that's human DNA, and I'm trying to cut out the gene of interest. I would have to use the same restriction enzyme to cut it out. And I can see the sticky ends of my gene of interest are complementary to the sticky ends generated by cutting the plasmid with the same restriction enzyme. So because bases are complementary to each other, where A pairs with T and G pairs with C, they will come together, and you can make sure the connection is permanent through using DNA ligase to glue the fragments together. Now that we have our recombinant DNA, this is our recombinant DNA from two different sources, we can transform these plasmids into bacterial cells. Remember the transformation, the term transformation is when cells take up DNA from the environment. So if I try to get these plasmids into the cells, what I can do next is have the cells grow on a petri dish containing ampicillin. If the bacteria did not take up the plasmid, they will be sensitive to penicillin and they will not survive. So only bacteria that have taken up the plasmid and have that ampicillin resistance gene will survive. Something else I see on the plate is I see two different colored colonies of the surviving bacteria. Some of them are white and some of them are blue. So what does that mean? It turns out, remember, that LAC-Z gene is disrupted when you insert your gene of interest into the cloning vector. So it turns out that plasmids that don't have the gene of interest will have an intact, a normal LAC-Z gene. And when they have this, they're able to metabolize a chemical called XGAL that's going to be on the Petri dish. When LAC-Z, that product, metabolizes XGAL, it produces blue colonies. So anytime you see a blue colony, that means you got the plasmid into the cell, but the plasmid looked like this on the top right, without the gene of interest. So you don't care about those. You want only plasmids with the gene of interest. Those are going to be the white colonies, because once you insert your gene of interest, you disrupt the LAC-Z gene, you cannot metabolize XGAL, and you don't see a blue color. So our next step would be to isolate the white colonies, and you can allow the cells to grow further to make sure every time the cell divides, you have multiple copies of the plasmid, and your bacterial cells can even express the genes to make your protein of interest. Besides molecular cloning, which is cloning a gene of interest, we can also clone cells, and cells do this themselves when they're single-celled organisms, such as bacteria. We know that bacteria divide through a process known as binary fission to create identical cells. Yeast can also divide through mitosis to create identical cells. Finally, we also have something known as reproductive cloning. And in reproductive cloning, we're actually making identical copies of whole organisms. So these are multicellular organisms, and this is a much more complicated process. In this process, what we do is we take an egg cell and we remove the nucleus from the egg. So if this is the egg, you take out the nucleus, and then you take a body cell, some kind of somatic cell, and you take the nucleus of the body cell and put it into the egg. And what we're going to see is this egg that has received a nucleus from a body cell, so it's a diploid nucleus, will ultimately be transplanted into another uterus so that it can develop into a fetus and to a whole other organism, and it will be a clone of whoever or whatever cell donated its nucleus. So the organism will be a clone of whatever organism the nucleus was taken from. Here we have a picture of Dolly the sheep, which is the first mammal to have ever been cloned. So how they took and created Dolly is they actually worked with 3 different sheep. And here we have the names of the different types of sheep from our textbook. We start with an egg donor. So this is the egg donor. It came from a sheep known as the Scottish black-faced sheep, and they took out the nucleus from this egg. Okay, so they enucleated the egg, which means you remove the nucleus. Then they went to a second type of sheep, and this one happens to be someone or a sheep known as Finn Dorset sheep. This one, you take their mammary cells, and they took out the nucleus from these mammary cells and transplanted that nucleus into the enucleated egg. So now you have the egg from sheep number 1 with the nucleus from sheep number 2. They took this egg, and now it's a diploid cell, and they actually implant it into a surrogate, a third sheep. And they let this cell develop into a zygote and then develop into a ball of cells known as a blastocyst, which is really a precursor to the fetus. And ultimately, we have Dolly as our final product. Based on what you saw from the previous slide, do you think Dolly, in terms of genetic cloning, is it a genetic clone of the Finn Dorset sheep or the Scottish black-faced sheep? What do you guys think? So this should be a clone of whoever donated the nucleus. So since the Finn Dorset sheep was the one that donated the nucleus, that is who Dolly was a genetic clone of. So many of the techniques we've been talking about are used in the field of genetic engineering, where we purposely alter an organism's genotype using these techniques from recombinant DNA technology. We usually do this through generating those molecular clones, where we clone a gene of interest and then eventually we try to introduce that gene into an organism to change its genotype. The organism that receives the recombinant DNA is going to be known as a genetically modified organism, and we often hear about this in our food in terms of crops, of our plants. If the foreign DNA came from a different species, that organism is known as a transgenic organism. For example, if we have something like corn that gets new genes from a bacterium, this would be a transgenic species because the corn got DNA from a different species. There's another subset of genetic engineering experiments known as gene targeting. And it's not really written this way in our book, but sometimes these are called knockout experiments. So back in the day when we didn't really know what each gene, the function of each gene, what each gene did in the body or what its purpose was, we used to randomly disable, not super randomly, but kind of randomly disable genes by mutating them or altering their gene expression in order to stop their normal function and look at the result in the body or in the cell. So when we target genes or go through gene targeting, we're disabling the genes to see what happens in the cell, and that kind of gives us a hint as to what the normal function of that gene or protein was. Closely related to these techniques is something known as genetic diagnosis. So what we can do is take a blood sample and sequence someone's DNA to ascertain a person or even an organism's likelihood of having a certain disease based on the different types of mutations or genetic markers that the patient has. Or if someone has already been diagnosed with a disease, we can study it further by sequencing their DNA because that might tell us the specific types of mutations that are present in their cells. As we saw in the previous chapter, talking about cancer, we can base treatment and prevention plans based on these results by targeting specific, for example, receptors in the cell that might have mutations. And these have generally led to much more effective therapies that target the specific disease that that patient has. Which specific mutations or changes do they have in their cells that we can treat specifically? Gene therapy is a genetic engineering technique that we use to try to cure disease. And here what we're trying to do is, in these diseases, the patient is missing a gene. And it's missing because either it's mutated or it's been deleted or it's somehow otherwise defective. In gene therapy, you're going to try to introduce a copy of the good gene into the cell in order to try to replace the bad version of the gene. So what we usually use in gene therapy are viruses. And here is an example. This one happens to be an adenovirus, which can be used to deliver the gene of interest into cells. An adenovirus can cause something like the common cold. So before we use this virus, we usually manipulate it to delete any part of the DNA that would cause disease, that would cause cold. So we get rid of disease portions of the gene, and we insert our gene of interest. So the gene that I'm trying to deliver into the cells to replace the missing gene. Then I use the virus, and I will have thousands of these viral particles with my gene of interest, and I infect my cells of interest. When viruses infect cells, and this is something we'll see in the next lecture, then they actually inject their DNA, and now the host cell, the cell that was missing the gene, will receive the healthy gene, and hopefully the enzymes will express these genes, and now you have replaced the faulty gene with a healthy version, and you might be able to cure the disease. Other common uses of these recombinant DNA technology techniques include making vaccines. For example, we have many different types of vaccines to fight viral and bacterial infections. Let's say I have a virus here, and this is something like a flu virus. I don't want to get sick from the flu, so I get vaccinated every year. What does that mean? What is the flu vaccine made of? This is a simplified version of this, but in essence, when you're making vaccines, what you want to do is take a piece of the flu, and I only take a part of the flu virus because I don't want the whole thing. The whole thing can make me sick when it's intact. But if I take just a piece of it, which I call an antigen, and I make many copies of this in the lab, I can inject the antigen into a patient's blood. That gives the patient's immune system enough of the shape of the flu virus to identify it as foreign and bad and attack it in case the whole intact flu virus is encountered in the future. So if we take genes from the flu virus, we can clone just that part of the virus, express the protein antigen, and use that as our vaccine. For antibiotics, this is just one example of what we can do, but we know that a fungus called Penicillium produces the antibiotic penicillin, but doesn't produce it in very large quantities. So in the lab, what we can do is cultivate this fungus and manipulate them so they produce a large amount of penicillin, way more than they usually make, and we can harvest that to use as our antibiotics. In terms of hormones, we can use molecular cloning in those plasmids to produce insulin or human growth hormone, because bacteria can take up those genes and express those proteins for us pretty readily. And then finally, this is not inclusive of all the common uses, but we can also use recombinant DNA technology to generate GMO foods. For example, one of the things that's pretty common is something known as BT corn. BT corn, as shown on the right, this is BT corn, and this is regular corn. BT corn contains a gene from bacteria. So this is from bacteria. And the bacterium name is known as Bacillus thuringiensis. You don't have to worry about that, but that's just where the letters come from. These bacteria make a toxin that's toxic to insects, but not to humans. So this is a type of pesticide resistance that you're delivering to the corn, because corn that has this gene can produce the toxin, and insects that try to eat it will die. Whereas regular corn do not produce Bt toxin, so insects are free to feed on this corn and damage it. So Chapter 17 is basically a giant list of common biotechnology and genetic engineering techniques. So our next one is mapping genomes, and for this one we have to know some vocab terms. The first one is genomics, which is the study of entire genomes, which includes the genes that are present in an organism, their nucleotide sequence and how they're organized, how these genes interact within the species and across species. In terms of genetic maps are just maps that tell you the location of genes on each chromosome. And this is through physical distance or genetic markers. When we look at genetic markers, these are maps that are based on linkage. So I don't know if you remember from our previous exam, we calculated things called recombination frequency percentages that tell us relatively how close two genes are to each other or how far apart they are. The further apart they are, the more likely they cross over. The closer they are together, the less likely they cross over. And then finally, we have physical maps. These are what it sounds like. It tells you the physical distance or location of these genes compared to other genes or markers. So again, we have two types of maps. We have genetic maps that tell us the relative location of genes in the genome, and this is based on recombination frequencies. So these are just estimates of how close or far apart genes are to each other. But then we also have physical maps that tell you the physical distance between genes. So you use different types of units for this one. One type of unit you could use is how many nucleotides two genes are apart from one another. Finally, the best type of mapping would really be based on sequence mapping, and we'll see this in the third video. When we have the genetic sequence of our DNA, we can have the most detailed information. Here's one example of a potential genetic map, and we've seen this picture before. Genetic maps tell us relative locations of genes. For example, A and B are relatively further apart from each other than B and C. And by calculating recombination frequencies that we did before, I will be able to see that B and C have a lower recombination frequency because they're so close together that they're less likely to cross over. Let's make up a number, let's say 15%. Whereas A and B might have a much higher recombination frequency because I can see they're further apart. Maybe they are like, let's say 42%. So if these are the recombination frequencies, what this genetic map tells me is the relative location of these genes from one another. Here's one example of a physical map. This is not the best type that we have, but it's the example from our book. So this is an example of a cytogenetic map. Here they have the chromosomes, they stain the chromosomes, and based on the appearance of the different genes or genetic markers, you can see physical distances from one gene to the next or one marker to the next. So here we're looking at physical distance between genes. All right, and that takes us to the end of our second video. In our third and final video, we're going to look at sequencing, genomics, and proteomics.